master's bachelor's degree from Ohio Northern University in mechanical engineering and master's and doctoral degree from Southern California also in mechanical engineering. He is interested in developing clinically relevant mathematical model of cancer, and he is also trying to develop agent-based model to understand the importance of spatial competition in cancer heterogeneity. He has many high-impact publications like Nature Communication, PNS, PLOS, Computational Biology, Cancer Research, and he is also very involved in science communication and co-editor of this mathematical oncology blog. So if you have any questions after this talk also, you can communicate with him. I think he will be helpful. And today he is going to talk on the topic of modeling cancer progression using agent-based methods. We would request Dr. West to share his screen and start the talk. Please, you can start now.
Thank you. Yeah, thank you so much for the introduction and the invitation. Okay, so I'm just sharing my screen now. Can you all see that?
Yes.
It should be okay, great. Yeah, so this is the title of my talk, and what I'm planning on doing with me, but the main reason I put that there is just because there is a lot of animations and things like that, so I don't want any sort of lag over the internet or anything like that to slow down our presentation here. So you're welcome to download that, and you're also welcome to tweet any of this or follow me on Twitter. Like Julie said, we're very active on Twitter and we use that for science communication. And later in the talk, I'm going to explain more about the Math Onco blog and how you might want to be involved in that. Okay, so I thought since this is maybe a bit broader and different audience than I'm used to speaking to, I thought I would start with some modeling philosophy at the beginning. And so this is kind of a fun, somewhat provocative quote about the nature of mathematical models. This comes from a review paper, which I think is very intriguing. And it says that models are not meant to be pathetic descriptions of nature. In other words, if we make a model, it's not supposed to showcase a very little pathetic piece of what we're observing in nature, but they're actually designed to be accurate descriptions of our little pathetic thinking about nature. So what does that mean? What that means is that our models come with a guarantee of accuracy. So that's what we do as mathematicians, as engineers. We can construct a mathematical model that looks like this. We list a set of assumptions that go into the model, and when it's published, we assume that the accuracy of the model is given. So that model is basically a way of translating assumptions into conclusions. Now, if we build the model hand in hand with biologists and cancer clinicians, then we're really building that model with a set of assumptions that is our current thinking about biology. And so how this is a little bit different way of thinking is the following. We can basically cast our assumptions about biology into the rigorous mathematical language. So if there is some fault with the model, we're assuming that the fault can be found with our pathetic, our little assumptions of biology at the current moment. This is really important for one main reason, is because this allows us to do something called forward modeling. Typically, you might be used to mathematical models that begin with a set of data, and we're trying to construct a model that might fit that set of data. Today, I'm going to be talking to you about a different style of modeling called forward modeling, where we begin with a known or suspected cause, and we express that in mathematical terms. And what we call this at Moffitt is hypothesis generating modeling. So instead of beginning with data and testing a hypothesis with our model, we generate a model and begin testing many different hypotheses. And what does that lead to? That usually leads to new experiments, right? So if a set of assumptions creates a model, and that model gives us some hypothesis, we should go test it in using more data, right? Leading to more experiments, more clinical trials. This is a really exciting time to be part of mathematical oncology, because there is a lot of encouragement and need for this style of modeling. So here's a recent review paper, perspective, and this was published, and there's so many great quotes from this. More theory is needed. It should be encouraged. We need mathematical models to implement new ideas. We don't just need to generate data, we need to generate ideas too. And these ideas will sustain our passion for mathematical oncology and oncology in general. Okay, so that's sort of all by way of background. And what we're going to do now is look at a hypothesis generating model. This one is looking at the role of tissue architecture. Feel free to interrupt me anytime with any questions. I'm happy to answer these as we go along. Okay, so this is based on published results. I'll share the paper in a second, but I want to sort of begin with the conclusion up front so that we don't miss it. So what we found using this model is that tissue architecture, which I'll go through in a minute, at tumor initiation governs the mode of evolution. And by mode of evolution, I mean either Darwinian evolution or neutral evolution. How did we get to that conclusion? We used an agent-based, or sometimes called an individual-based model, operating on a square lattice in explicit space. And then the approach that we took to finding that conclusion was that we systematically alter the spatial constraints and cellular mixing in that math model. And we're going to apply that to a type of breast cancer, ductal carcinoma in situ. So if you're not familiar with Darwinian or neutral evolution, a picture is shown on the right here. And what we have here is tumor initiation in blue. And we see that blue clone expanding, having many daughter cells. And then there's a mutation to the red clone, which overtakes the blue clone. And then the green clone overtakes the red clone, and so on and so forth. These subsequently more aggressive clones are sweeping through the population. That's called Darwinian evolution, or clonal sweeping. And on the bottom here is where we have many coexisting neutral clones. So there's no sweeping that occurs. They all coexist at the same time. And that's a more neutral evolution. In other words, a neutral competition between clones. So when I was constructing this model, I looked back in history and looked at some of the literature of agent-based models that made a big clinical impact. And so this is sort of a mini timeline of these simplified cancer models that are called passenger-driver models. And basically what happens is you have two types of mutations in this model: a driver, which increases the birth rate, or a passenger mutation. And it's called a passenger because it's carried along for the ride. Usually they're considered to be neutral. They might be slightly deleterious. So why would a cancer cell have a passenger mutation that's slightly deleterious? Well, it's because they're carried along, their passengers are carried along by drivers. So the drivers are the drivers of evolution, and they may carry along these passenger mutations along the way, even though they may be slightly deleterious in a Darwinian fitness sense. So we want to present an extension of this model, which includes tissue architecture. And what I mean by tissue architecture is the role of space. All of these previous models were typically non-spatial models with one or two exceptions. But what we want to do is get more accurate into looking at the actual architecture of a ductal carcinoma. So let's start with a very, very basic model or basic simulation here. We have different cancer cells, which are in colors here, on a grid. And so the white locations are empty locations. And this cancer cell has one driver, case of D, and two passenger mutations. And we may have a mutation that occurs at some point in time, and this has two drivers and the same two passengers as before. So we see the simulation with some amount of cells in the middle of the simulation, and then we let that grow out. And you can see that a mutation occurs in red here, and that red clone eventually overtakes the entire population. It sweeps through the population. And then it's another mutation, and that mutation sweeps through the entire population, and so on and so forth. So the logical question we can ask is, what is the role of spatial constraints? So we repeat this simulation with different domain sizes. And the result is very simple here. You can see that in general, these smaller domains are typically homogeneous. So if you glance at it at any given point in time, you see that it's typically one color, right? Compare that to the right, where there's many different colors, many different clones, a very heterogeneous population. And so maybe I'll run that one more time. So basically we see that smaller domains increase the rate of clonal sweeping across the population. But you may say, well, this isn't a fair comparison because these domains are all different sizes. So of course the bigger one is going to have more clones. So let's do a similar simulation that looks at the same domain size, but we segregate that into different sizes of segregated domains. So in this case, you can see that the single region case is again more homogeneous. There's less number of clones here because each of those clones can sweep through the entire domain. And on the right-hand side, you can see that it's a very heterogeneous collection, and each of those individual regions is very homogeneous. So this segregation increases the global heterogeneity across the whole domain. You can see this whole domain is practically orange, while this domain on the right is many different colors. But the individual regions within that domain are very homogeneous, right? So that leads to the interesting observation. What if we see these simulations from a single region and we allow for some rate of dispersal between regions?
Okay, so this is an assumption in the model. I'm going to explain how this is rooted in biology in a second. But let's just look at the dispersal rate between these two. So here we have a very low dispersal rate. And you can see that before that first dispersal event occurs, we actually get some sweeping in the first region before we seed a new cell into the neighboring region. Compared to this case, where the dispersal is happening so quickly that limited clonal sweeps are occurring in that first region in the middle. So let's quantify this a little bit. We're going to quantify this on two axes, mutation burden and the average number of drivers. So what I'm going to tell you is that the right or the x-axis represents some level of neutral evolution, and the y-axis represents Darwinian evolution. That's the number of drivers that have swept through the population. So you can see in black here, this is the single region case. In black, a lot of clones are developed, and then we see a upward turn on the trajectory towards Darwinian evolution, and then a shift to the right again. Compare that to the green case here, where we see an immediate shift upwards, acquiring many drivers. And you can see this in the simulation. At the beginning, you go from blue to red, right? But in the left with one region, that blue to red transition happens when the tumor is already fairly large. Compare that to the right-hand case where that blue to red shift happens in that first region before the dispersal event, and making that the tumor is very small when it acquires that next aggressive mutation. What happens when I increase the dispersal rate? All of these curves collapse back onto each other. That means that this segregation, those boundaries, don't have any effect anymore. This makes sense because any spatial constraints imposed by those boundaries is lost if the cells are allowed to disperse at a high rate. So the summary of this slide is really that this low dispersal rate combined with these high rates of segregation of spatial constraints enables a more Darwinian evolution. So I wanted to go back and look at each of these cases again, but looking at passenger mutations. So we can compare the same simulations as before, these driver mutations, and then on the bottom we have passenger mutations. So we do see an example of this passenger-like behavior. You can see down here, there's an event of this passenger mutation sweeping through the population. Well, I've already told you that passengers are deleterious, so why would that sweep through the population? Well, it's because whichever driver was sweeping through the population here is the one that carried these passenger mutations along. So you can see that the purple clone relatively matches this sweeping population down here. And so that's when we're talking about mutation burden, we're talking about basically the sum of both passenger and driver mutations here. Okay, so we can look at the same thing for our other case and look at the different passenger mutations. And we see that in general, it follows a similar trend, that it's more homogeneous on the left here and more heterogeneous on the right. And so let's look at our mixing case as well. Again, it follows a similar trend here as well. So we can see that basically these smaller region cases are what accelerates evolution. Okay, so what have we learned so far? really simply, we learned that smaller domains increase our level of clonal sweeping. These smaller domains are sweeping through the clone more easily. And more domains increase our heterogeneity. But sort of the combination of those two factors is that some limited dispersal between domains accelerates the acquisition of new clones. That's because smaller domains increase clonal sweeping, and if you allow for dispersal into new unexplored territory, that aggressive clone can expand. So what I'm saying basically is that this is an example of natural selection in action. These harsh spatial constraints are selective, and it's selecting for increased aggressiveness of driver mutations. What does this all have to do with breast cancer? So you guys have indulged me by looking at a simulation which so far doesn't have too much grounding in reality. But right now what we're going to look at is its biological application. So these are ductal network structures that are from this paper called breast computed tomography. And basically, ductal carcinoma in situ is constrained in these ductal network structures. This is a pre-invasive cancer before invasion into the surrounding tissue. So depending on the point of tumor initiation, where you initiate the cancer inside of that ductal network, you will be subject to different spatial constraints, right? Some of these networks are smaller, some of them are larger. You will also be subject to different dispersal constraints. Depending on the exact tomography and the branching, that two different regions of the tumor may be separate, even though they're physically close together, they may be separated into different branches, and dispersal between those branches may be difficult. So if we take a scan of one of those ductal networks, we can see that basically at the top here, we have very small branches and very many branches. And at the bottom, it's the opposite, fewer and larger branches. So we take that same model and simulate it inside of that ductal tree, and we can see the evolution of drivers and passengers in this biologically relevant tissue architecture. And so what we do is we plot that result on the same graph that we had before, where we have our Darwinian versus neutral evolution state space, so to speak. And what we find is that if we initiate in those larger ductal branches near the root of the tree on the bottom, that we get sort of a slow and steady slope on this state space. But if we initiate it closer to the top in a more tightly spatially constrained branch, we get this vertical evolution characterized by Darwinian evolution. And what we're saying is basically this model shows us that we can track the evolution by tracking the angle or the slope of that curve at any point on that state space. What's really interesting is that it's this green curve starts out very Darwinian, and then it takes a sharp turn towards neutral evolution. And there is some evidence to support that in the literature. Basically this kind of a structure in tumor evolution. Early in tumor initiation, we get lots of clonal sweeps, and then it eventually merges into a more neutral style of evolution. So this is our summary schematic that we showed before. And basically, we can say that the point of tumor initiation, either in these large branches or small branches, governs the mode of evolution from Darwinian on the top to neutral on the bottom. So can we match this to some clinical data? In particular, what we want to look at is a clinically relevant metric of evolution. And there's something called the variant allele frequency, where basically you look at the different mutations in the tumor and how often those mutations occur, the frequency of those mutations. And then we plot the frequency, 1 over the frequency, as a function of the cumulative number of mutations. And there is a theory that says that the relationship between these two variables is going to be linear if the tumor is neutrally evolving. So if you're interested in this, I would encourage you to read this paper. It's a really nice explanation of this theory. But what's interesting about the paper is that they speculate that these neutral evolutionary dynamics may be influenced by the architecture, the tissue architecture, how these clones are competing in space. So let's take that same measurement and just look at it over time. So this is our 1 over F metric. And I should say that, that if the 1 over F metric approaches a value of 1, then that's considered neutral evolution. So we can see that early in our simulation, these tumors are not considered to be neutral. There's a lot of clonal sweeping going on. But later in evolution, these are considered to be neutral because they're approaching 1. This is when we might observe these tumors clinically, so we can sort of see where that paper was coming from that is classifying all of these tumors as neutral, because it's likely that we're measuring it at later times when it presents in the clinic. But we can show the same sort of trend where we change the point of initiation of our tumor. And you can see that the green, that was the more aggressive Darwinian tumor from before. The green kind of hangs out at the bottom here in the Darwinian mode for longer before eventually approaching this neutral evolution. So basically, all the tumors eventually go to neutral, but if you have a higher spatial constraints in the beginning, it allows you to be Darwinian for a longer period of time and eventually be more aggressive even in later periods of time. Okay, so I'm at a transition point. So if you have any questions about that project, now would be a good time to ask them. If not, I'm going to sort of showcase another second example of agent-based modeling in cancer.
Hi, Jeffrey. I had a question.
Sure, go ahead.
It's about you said that the clones show Darwinian or neutral evolution based on the space constraints and dispersal, if you allow it, right? So, do the same clones show both types of evolutions based on how you put them?
Yeah, that's a great question. So one thing I will say is that this measure of Darwinian evolution is on the tissue scale. So it's looking globally, right? So that's a bit of a tricky question because each of these individual clones, like say the yellow clone, it is associated with a number of drivers. But what actually changes the nature of the evolution is what that clone is interacting with. So you can see that this yellow clone is interacting with the red and the green and the blue here. So even though that clone may be still operating according to Darwinian principles, its surrounding cellular neighbors cause it to act more neutrally in comparison with its other clones. Does this answer your question?
Yeah, it sort of explained it, yes.
Yeah, it's a complicated thing. So what we say is the cell-specific characteristics are constant. So the cells are behaving the same way that they would, but the tissue-specific characteristics are what are evolving over time here.
Right. Okay. Thank you.
I have one question. Like, what
Sure.
kind of computer programs that you need to learn to do this? Like, if our students they are interested, then what kind of programs they should learn a little bit so that they can in future maybe they want to pursue this, then that will be their like in advantage.
Yeah, that's a great question. I should have put a slide in here. Let me see if I have one at the end. So what this was done with is something called HAL, a hybrid automata library, and this is written in Java. We have a lot of tutorials for this sort of agent-based approach if you're interested. And in fact, this particular model that I explained today, I have a tutorial on the Mathematical Oncology blog. So I think, yeah, I might as well show that now. So if you go to this link, I actually do have a tutorial on this exact project that you can visualize evolution of passenger and driver mutations over time. And I'm happy to follow up with you all afterwards if this is something that's interesting to you to get started. So we've tried to make this process as easy as possible to sort of come into mathematical oncology as a field and get started right away. So there are a lot of exciting open source tools for you to use. And I should have put the link in here, but if you go to this website, you can search by author. And if you search for my name, it should show a tumor evolution tutorial in there.
Yeah, that's very nice. Yeah, thank you.
Thank you. Okay, so I guess I'll move on to the next section of the talk. And this is a totally different model, but what's consistent is that it's an agent-based model. I like to explain this model in the context of a chess metaphor. I love playing chess, maybe some of you do too. And so I'm going to sort of explain this cancer model in context of a chess gambit. And for those of you that don't know, a gambit is essentially a chess technique where you sacrifice one of your pieces, typically a pawn, with the hope of achieving a resulting advantageous position. So in this case, you can see that the white pieces are sacrificing their white pawn here on c4, and black captures that pawn. But the idea is that white gains some advantage through that sacrifice. This metaphor came about by looking at a tweet by one of my colleagues at Moffitt, who asked, if you asked a tumor its opinion on the immune system, what would it answer? So there's 4 choices here. The first choice would be a worthy enemy. In other words, maybe the immune system is very successful at targeting new tumors, but we just don't see much evidence of that. So maybe there is some mutation in our body and our immune system takes care of that. So without our immune system, we would have much, much more cancer. But with our immune system, it can eliminate all but the most aggressive tumors. So that immune system is a worthy enemy towards most tumors. Or maybe it's somewhere in the middle. It's not my favorite, but I can deal with it. Or here's the most popular answer, is that the immune system is a pawn in my evil plan. In other words, the tumor can co-opt the immune system for its own advantage. Or maybe the last answer would be, what immune system? Maybe the immune system is just not involved in cancer evolution. So what we're going to explore is this third answer. So I need to introduce the model before we get into our gambit metaphor again. This is a much more complicated model, and these are the relevant publications here before my time at Moffitt. This is a model of cancer metabolism where we have many different cell types. Before, we only had one cell type, which was cancer. And here we have normal cells in the gray in the background here. And we have white cells that represent blood vessels. And we have tumor cells, which are shades of color here. And we can also have necrosis. And these all operate according to some cellular rules in the model, which I'm going to sort of gloss over and refer you to the paper. Okay, but each of those tumor cells has 3 different phenotype axes. We can evolve acid resistance, which is just resistant to acidic conditions, or we can evolve increased glycolysis, which means that we can divide faster. This is a characteristic of cancer cells, rapid division. And we can evolve PD-L1, which is an immune biomarker, which enables cancer cells to hide from immune T cells. And in the model, we also have some diffusible factors. So this is called a hybrid model because we have the agent-based components, but we also have PDE components, differential equations describing oxygen, glucose, and acid. And what I've added to the model recently from past publications is the role of the immune system. So these are T cells, which are immune cells in orange here, which swarm in and target the tumor and kill them. So it basically kills tumor cells. And there's some methods for the tumor to escape the immune system, which we'll get into in a second. So this is a very complicated model. In fact, this is all of the elements of the model. We have our tumor grid here, and then we have our phenotypes, acid resistance and glycolysis here. And then we have some vasculature parameters, some T cells in blue, and then we have our microenvironmental conditions, oxygen, glucose, and acid or pH. And then we have some immune escape mechanisms, that PD-L1 axis of hiding from the immune system. And T cells can also be inactivated in very acidic conditions. So in this region of high pH, we won't see any T cell attacking the tumor because T cells are inactivated in highly acidic regions. So we can see that our tumor phenotypes evolve over time, and I'm going to go into more detail in this later. But basically, at the end of the simulation, we see this very aggressive top right phenotype, which is associated with our increased glycolysis here and increased acid resistance. And that's called our aggressive phenotype. So just think of this pink phenotype as very aggressive, invasive phenotype. And we can also track in the model the susceptibility to the immune system over time. And we can see that in red, it's very susceptible. And as the immune system targets it, the tumor actually shrinks. And then it develops some escape mechanism to avoid the immune system. And the tumor becomes very white, and where the immune system has no chance of targeting that tumor. So we're going to sort of investigate some immune escape mechanisms. But before we do that, we need to sort of get a baseline feel of our simulation without any T cells in it. So here's an example simulation where you can see that there's phenotypic evolution that occurs. Some of the cells evolve acid resistance first by moving up to the top to that blue phenotype, and then eventually over to that pink phenotype. So let's look at this a little bit more closely. What we do is we see the tumor in sort of like a donut shape, and the center of the donut is a very harsh condition. Again, this is very reminiscent to ductal carcinoma in situ, where growing into that empty space in the duct represents growing into a very harsh acidic condition. So you can see that the acid in the center of the tumor is very, very high in blue. It's a very acidic core. And what happens is that acid core is essentially a factory of evolution. These cells are dying at a higher rate because of harsh conditions, but that increases the selection in the tumor. And that is where these aggressive clones are produced in that factory of evolution. And then they have to make their way out towards the normal tissue to be invasive. So what happens is that a vessel collapses, which is a normal event in our bodies. Vasculature is renewing. And then these aggressive clones sort of have a way of escape into the surrounding tissue, and they can become increasingly invasive. So now they're proliferating into nice conditions. They went from the harsh core to the nice normal tissue surrounding the tumor. And then what happens after that invasion is this pink phenotype may emerge and become increasingly invasive. And that is called a Warburg phenotype. So you may hear me refer to that in the next few slides, this Warburg phenotype. So this is, I think, the, yes, so why is immune predation important? If we were going to construct a mathematical model of immune predation, why would we use this one? Right? This is a cancer metabolism model. How does metabolism interact with the immune system? Well, a couple different ways. I already mentioned that T cells can be inactivated in high acid microenvironments. Also, T cells can compete for glucose with cancer cells. Cancer cells consume a lot of glucose, and this is basically an immune escape mechanism of starving the T cells of glucose. And the T cells are recruited through vasculature. So if a region of the tumor is less vascularized, then we would expect it to have less immune predation too. So here's what happens when we add T cells to the simulation. And here's where we get back to our chess metaphor. We can see that in the no immune predation, we have this nice green rim of essentially metabolically normal, not aggressive cells that trap that factory of evolution in the center. But when we add T cells, the T cells come from the outside and they target those green cells first, eating away at the tumor. Well, that enables the aggressive cells on the center of the tumor to invade into that surrounding tissue even more easily. So that's really interesting because it reminds me of that pawn gambit that we saw in the beginning. So never underestimate the power of a pawn, because we may sacrifice that pawn. The tumor may sacrifice those green cells in order to gain a positional advantage where this pink phenotype emerges more easily. Okay, so this is a bit of a diversion, but I'm going to sort of explain how we might visualize this model. So we're going to use something called phenotypic barcoding, which I think is really cool. And also it's another one of the open source tools that we have available to you if you want to sort of use this in your own models. So sometimes we use what's called a Mueller plot to visualize evolution over time. I already sort of alluded to one in the previous section of the talk, but basically what this is showing is clone size on the y-axis over time. And we start here with 4 different cells, and we can see that this third cell is the one that has many, many daughter cells and eventually expands to consist of most of the tumor population. And these 2 clones die out, and then this one is coexisting, right? So what if we took at a later point in time a biopsy of the tumor, and then we counted the number of cells here, and now there's 10 cells, and we can see that these 6 cells belong to this third initial cell, and then these 4 cells belong to this first initial cell. And since each of those 10 cells may have acquired new traits during the simulation, we can recolor those 10 cells the current phenotype that are associated with those 10 cells. So we essentially color these new colors, right? But we preserve the lineage information. So these cells came from this green phenotype, but they have drifted through phenotypic drift to new phenotypes. So we can do that again. We can recolor the subsequent evolution over time. And this is important because we can show the phenotype as well as preserving the lineage. So it's essentially barcoding over time. We take a sort of a barcode and tag each cell with an individual number, right? 1, 2, 3, 4. And then it carries on its original number in red here, but it has a new barcode at the second time point. And we can determine those phenotypes over time, and it's a nice visualization. So this is how we produce these plots on the bottom here. We can showcase both the lineage information as well as the phenotype information over time. So this I think is really, really cool. And again, I'm happy to follow up with you to sort of show you how this works at another time. We have a blog post on that topic as well. Okay, so as we're finishing up the hour here, I'm going to sort of show some results from this model. One of the key parameters in this model is vasculature. So we're going to look at the role of vasculature. In particular, we have two different parameters. One is the stability. Remember I said that a vessel could collapse. That collapsing happens at some number of days on average. And once a vessel collapses, it's ready for a new vessel to emerge. And the other parameter in the model is the probability of a new vessel emerging. So we're going to look at two cases that are clinically relevant sort of verbiage describing these two different parameter sets. The first is intermittent hypoxia, that the vessels collapsed after a very few amount of days. They collapse very often, but they're renewed pretty quickly. And then the second is weak vasculature. Vessels collapse very often and they're not renewed very often. So that would be associated with very harsh conditions. So on these same two axes, we see the evolution of these phenotypes over time. And you can see here that larger tumors in white typically are associated with this weak vasculature case in the bottom left. It's also associated with highly acid resistant in blue and this more purplish pink Warburg phenotype. So in other words, harsh vasculature conditions select for aggressive phenotypes, and those tumors grow faster. Here's an example of those two cases. This is what I was talking about, this weak vasculature being more aggressive. So you can see over time that this pink phenotype emerges very quickly. All things being equal except for vasculature, this case grows into the center and the vasculature is replaced right away. So that center becomes well vascularized. You can see these little white blood vessels filling the tumor evenly, basically. And so less evolution occurs over time. So we'll keep that in our minds that basically increasing our vasculature settings allows us to slow down evolution. Let's look at these same two cases with and without immune response. So on the top here is no immune response. We have the aggressive case and the non-aggressive case. In the aggressive case, as we add more and more T cells targeting that tumor, the tumor just shrinks. It's a monotonically shrinking scenario where more T cells always kills more tumor cells. On the right, however, if we add T cells, we can actually see that we select for increased evolution. So where there was very little evolution occurring without T cells, now there's a lot of evolution occurring with T cells. So we can call that an immune-mediated bottleneck. Essentially, the immune cells are the selection pressure acting on the tumor, and it's encouraging evolution to occur where it might not otherwise. And another way of looking at this is plotting the T cell recruitment rate. And what I said before is this weak vasculature case is monotonically decreasing. So if you have T cells and you're going to inject them into the tumor, for example, then you would always want to inject more and more T cells because it's always beneficial. But contrast that with intermittent vasculature, we have this middle range where we can actually do damage to the patient by recruiting more immune predation because we're selecting for evolution that wasn't previously there. So in other words, we could think of that case as a fairly benign tumor, and that immune system is selecting for the aggressive traits of that otherwise benign tumor. And again, this is you can see where the gambit comes in here. This is just to show that the phenotypes are also being selected for here. So that was looking at the growth rate. This is looking at the reason behind that, which is our aggressive phenotypes. Okay, so in the last few slides, I'm just going to look at some quick treatment implications. And we have two treatments here, anti-PD-L1, which is an immunotherapy, and buffer, which targets acidic conditions in the tumor. So I said before that our PD-L1 is a phenotype which allows the cancer cells to evade the T cells. And anti-PD-L1 is a drug that basically reawakens the immune system to be able to target those cancer cells again. So what is the effect of anti-PD-L1? Essentially, if you look at this untreated condition here, this is what we had before. It's a different simulation, but basically we have aggressive evolution and fairly benign evolution. This one does have some aggressive clones emerging towards the simulation because I just ran this one for longer periods of time. But even so, comparatively speaking, this is still the more aggressive one. If we were to add anti-PD-L1, essentially that's reawakening those immune cells and increasing the immune recruitment. So what we would expect is that in the left case, immune recruitment is always good, right? Always beneficial. So we can't go wrong adding anti-PD-L1. But in this case, we would expect that we can go wrong adding anti-PD-L1. And we see that bottleneck occurring and this more aggressive phenotypes emerging more quickly after anti-PD-L1. So the other drug we have is buffer, which essentially eliminates acidic conditions within the tumor. And that slows evolution of acid resistance. So you can see that in both of those cases, it has a marked effect on the evolution of a tumor over time. And we can also look at the timing of those different treatments. And what we find is generally in both cases, the blue representing buffer should be given early and the anti-PD-L1 should be given late. So we get a nice treatment implication from our model of when to give treatment and how or what the vasculature setting or parameters should be to sort of guide treatment implications too. Okay, I think I showed this slide before, but again, happy to follow up with anybody if they'd like to get started on some of these tools. To visualize those phenotypic barcoding, I use EvoFreak, which is an R package, but we've created a package in HAL to basically export these tumor evolution plots in a few lines of code to be visualized in EvoFreak. So if you want to make basically diagrams like this, then it's really easy to do in HAL plus EvoFreak. And then this metabolism model is previously published as well. So I'd like to sort of end my talks describing some of the resources that we have available that I would love to be able to share with you all. This is our website on Math Oncology, and we have a couple different resources on that website. So I would encourage you to check this out. It's mathematical-oncology.org. We send out a weekly newsletter of publications and blog posts. So if you'd like to subscribe to that newsletter, It's called This Week in Math Onco. And we have recently reached the number of 1,000 subscribers. So if you publish something relevant to math oncology, definitely send me an email, and I'm happy to share that in the newsletter with our subscribers to this newsletter. Basically, we just post preprints and publications, and it's a way of building community around these mathematical oncology techniques. And that can be found at the same website /newsletter. And then the last resource that I mentioned before was the blog. This would be a great place for any of you to submit a blog post. We're happy to have new authors on the blog. We've had, I think, over 40 different authors. So this is meant to be a community resource where you can write a post describing a recent paper or write a tutorial like I've done. Definitely, we're willing to solicit submissions to the blog, or you can go back and search through some of those and look for tutorials and things like that. With that, I'd like to acknowledge and thank my collaborators. And again, you can feel free to contact me on Twitter and email, and happy to take any questions. Thank you for listening.
Yeah, that was an awesome talk, and we learned many things that like how to do this agent-based modeling and how to see that whether the cells are interacting or not. So I have one or two questions. Maybe the students also having some questions. So I'll start. First, I have two questions. Like when you are doing the modeling of this tumor formation that you are saying, uh, is there any specific numbers, like what is the cell number, or is there anything that you need to take care of? Like you start with a certain population or something. And second is like when you are saying that the simulation is going for 150 days. So is it real time 150 days simulation, or like it's I don't know, like how you calculate these things, or is it
Yeah,
real time?
that's a great question. So I think, yeah, broadly speaking, it's difficult to develop an accurate parameterization of agent-based approaches, especially in evolutionary approaches, because things like mutation rates or phenotypic drift rates are difficult to measure in oncology, especially. So how can we sort of match our model to data? I think there's two answers to that question. The first answer is we can use mouse models to map some of these parameters to data. So these different papers kind of go into the more details of how the specific parameters were chosen. I sort of skipped over that part because I like the second answer to this question, which is what we can do is sort of look at a range of parameters and just see their relative qualitative effect. So this is less about predicting patient-specific dynamics and more about looking at qualitative differences between various parameterizations. So usually we take a wide range and then just look at the full range of dynamical behavior that's possible, rather than trying to narrow it down to one specific parameter set that we think is accurate. We'll just see different trends. And the idea behind that is the philosophy of modeling I explained at the beginning, of just trying to develop new hypotheses to go back to the lab and do experiments on. So I hope that's a satisfactory answer to your question. It's definitely a different style of modeling.
Yeah, when we do like the cell death rate or the division rate, this all will change depending on like what is your start value or what is the start point. So I think obviously these are, you are looking like you are considering them because you are seeing that the cell death and nutrient metabolism or the nutrient availability will be all connected. So this will probably like for our, because we are mostly wet lab people, so for us, these things, these are important points like we need to think about. And my second questions will be like, here we are looking into the T cells, like they are killing the tumor cells. Like
Mhm.
you are looking into that one-way interactions that the T cells are killing. But
Mhm.
in reality, there could be some sort of an crosstalk that the T cells are not killing them, and they are helping them. Like when we see macrophages and all.
Mhm.
So, do you think that kind of approach can be add to this kind of story, like the T cells not killing them, but they are helping in different way,
Yeah, that's a great
not being the
question.
pawn, maybe being
Being the coach or something. Yeah.
yeah.
um, yeah, no, that's a great question. I think that's something we would like to add to the model. The co-evolution of T cells and tumor cells. Tumor cells can feed back on the T cells. Right now, they're essentially, the T cells are really dumb in the model. They're just kind of randomly wandering killer cells. But we know that the T cells can be co-opted or co-evolve alongside the tumor. So we've ignored that for now, but I think that's a great thing in the future. Especially there's some data on sequencing T cell population in patients to sort of get a hint at what those T cells were targeting in the recent past. So we may be able to sort of parameterize some of these things from data, from patient data in the future too.
Okay, that's nice. Any students, you have any questions?
Yes, I had a question.
Mm-hmm.
You told us about your tutorial on the blog. So would that be a good starting point for a complete beginner in mathematical oncology?
Yeah, I think so. Yeah, maybe I can just pull that up since you asked about it. If you go to mathoncology.org and then the blog post here, if you go to authors, and then if you search my name on the bottom here, Jeffrey, and then there's one called How plus Evo Freak. So I think this would be a good place to start because it actually has some code in here too to visualize these Evo Freak plots. And HAL is basically our library in Java to be able to do the agent-based simulations, and it's optimized for speed and visualization. So all of the movies and things like that that I showed before, that's all HAL. And then anytime you want to visualize that over time, then that's Evo Freak. And so you can see in this blog post, there's some code and description and stuff.
Okay.
So, and definitely don't be afraid to email me. I'm happy to answer questions about this. I would love to interact and meet.
Yes, definitely. Thank you.
Thank you.
Any other
And
questions? Yeah.
yeah, I just had one more. Sorry. Uh, is any of the simulation done in Python?
Oh,
Like,
that's a good question. Yeah. I think some people use Python packages for agent-based simulation. That would be fine. I personally haven't worked with it too much, but some other people at Moffitt I know do. So that's a
okay.
great language for that. There's many
Yeah.
resources out there.
Yeah, I asked since it's a pretty well-known language now, so
Yeah,
I figured
that's right.
someone might be working on that.
Yeah, that's a good question. Nothing comes to mind right away, but yeah, I'm sure you can find some resources.
Yeah. Yes.
Hello.
Go ahead. Yeah.
Yeah. So it's like when you design a mathematical model, so is it like you have a hypothesis and then you make the model and then you test it on real samples? Or is it like you do a study and then you run a model that will fit into that?
That's a great question. I think both are appropriate. the reason why I introduced sort of hypothesis generating this forward modeling is because I think it's less common.
Okay.
The places where this really shines, in my opinion, is where classically data is difficult to be collected. So for example, in that second model that we looked at, phenotypic evolution, do we know how fast individual cells acquire acid-resistant traits? That's a difficult thing to measure.
Yeah.
So, you have to sort of give your best educated guess at that parameter in the model, no matter what. Maybe we'll be able to measure it in the future. And you may be able to measure it with some sort of data saying we can put boundaries on that parameter based on the rates of tumor growth or, something like that. But the other way of doing it is just, well, let's look at all the parameters and see what are trends. And the useful thing about trends is basically that it gains some intuition about the biological system. And when we show that to our biologist collaborators, that may spark an idea for a new experiment.
Yeah. Okay. Thank you.
That was a great question.
So I wanted to ask you that how do you incorporate the biological data? I mean, right now people are doing next generation sequencing and sort of things, or even different type of microscopic techniques. So how do you convert that data into mathematical assumptions, and how that is used for sort of forward modeling?
Yeah, that's a great question too. So let's see. So in the first part of the talk, we're talking about genetics, right? In genetics modeling, in genetic sequencing, there's a lot of what are called summary statistics. I mentioned one of them, which is the 1/F summary statistic. This 1/F statistic in patient data, this might represent, 1 million or several million cells sequenced, right? In our simulation, maybe we'll only have 10,000 cells. So we will never sort of scale these simulation to the full number of cells. But one way of matching to data, especially genetic sequencing data, is matching these summary statistics. So it's an aggregate value of the whole population. And if we run many different stochastic simulations and average those summary statistics, then we can see and match that back to patient data. So rather matching the individual genetics of specific cells, we just match these summary statistics of, the average value across many simulations. In the metabolism model, it's probably a little bit more complicated. They use some mouse models, and then they have ways of measuring some of the phenotypes, but not all, or sequencing the tumors after some period of time, or looking at the growth rates, and then trying to match very specific parameters. But some of the parameters are more tricky to measure than others.
Okay. So if we begin with a very simple in vitro model consisting of a homogeneous type of cells, and then we have some sort of macroscopic data where the cells are changing their morphology and showing some different morphological phenotype. So how can we convert these data into some mathematical model and then interpret what is happening?
Yeah, that's a great question too. So when you mean like what kind of, you said morphological phenotype, is that what you said?
So yes, so if in our model, if the cells are first in the beginning, the cells are looking circular, and then if they are changing their morphology into some other like maybe they are getting spindle shaped by after some time or after some treatment. So how can we convert this data into some mathematical model and then maybe predict what can happen?
Yeah, that's great. That question, I have seen papers looking at mechanical, forces in cell structure and changing over time.
Yes, yes.
I haven't really worked with that too much in the past, to be honest. So this hybrid automata library, HAL, we typically don't do mechanical force models in that. So there are other libraries that would be good for that, like Physicell, for example, where they're actually looking at physical forces and interacting cells over time. And you could sort of account for morphological changes. And I'm sure there's other packages too. But I don't have any experience in that. So it would be really cool to learn some of that too.
Okay, so maybe a particular type of data is required to generate a particular type of mathematical model.
That's true too. It depends on your question. So since I work in a cancer institute, we try to let our model questions be dictated by clinicians and biologists. What clinically relevant question do we want to answer? And then a lot of times it starts with a hypothesis. The clinician says, I have this hypothesis about, the genetic makeup or evolution of a particular tumor type. Can you build a model of that? And then we'll build a model of that and see if it matches their intuition. And the model should typically be as simple as possible to capture that set of assumptions that the clinician originally arrives at. So yeah, that's a very vague answer to a complicated question. So I think some models, like you can see that in this first part of the talk, the model is so simple, one cell type, two different types of mutations. And in the second part of the talk, the model is so complicated. So we can answer a more complicated question. But I'm not against simple models at all, because if this was a very specific question, we don't want any other complicating factors to influence the answer to that question.
Yes.
Yeah, hello. I have one small question.
Go ahead. Yeah.
Yeah. So this is very nice. I mean, your talk was so good. One thing I'd like to know if this can be used to personalize treatments, and what would be the minimum data required from the patient to be able to do something like that. So deciding a personalized treatment regime or something like that.
Oh, I love that. Yeah. So yeah, I wish we had time to do another talk, because that would be a great question to sort of launch off into a whole new talk. But because the other area of my research is adaptive therapy, which is personalized cancer treatment. So I'll just make one point here, and that's that if you want to build predictive models, that's going to be a little bit different than descriptive models, let's call them. So predictive models are, to predict patient-specific dynamics. So usually I would say those models are done in ODEs, differential equations, and that's mainly because the stochasticity of these agent-based models makes it difficult to build a patient-specific predictive model from them. We can identify trends from these models and act on them and design new experiments or trials from that. But if we want to actually do feed-forward prediction of patient dynamics, then we'll probably use differential equations. I think you asked, like, what's the minimum amount of data to sort of drive these decision making. The current status of this is really limited right now. There are several examples of mathematical models which are the main drivers of clinical trials, but it's very few. I think there's only maybe 2 or 3 examples of this, where basically the patient dynamics are fit to a math model, and then a new treatment decision is made based on the math model. So the field is really at the tip of the iceberg in trying to do that. The exception to that is a lot of toxicity sort of development of new drugs and determining the toxicity profiles of a particular drug for patients is done with math models in pharma a lot these days. But actually driving the treatment protocols is more difficult to consider evolution and resistance and those kinds of things. But that's one thing we're interested at Moffitt. So if you sort of look into adaptive therapy, that we do this in prostate cancer, and the nice thing about prostate cancer is we do have a biomarker of tumor size over time. And that biomarker is the blood draw, and we take basically a measurement of the blood to predict the tumor size over time. So we have a nice dynamical profile of time-dependent evolution. And some people are also doing this in imaging, but analyzing images has to be fast enough to keep up with patient data too. So this is like a huge, huge problem that a lot of effort is being put into from my point of view.
Right. Thank you.
Yeah, great question.
Hello. Hi.
Hello. Yeah, go ahead.
So I had a question. Can immunotherapy, which actually help the immune system to fight cancer, be sometimes harmful in case if the aggressive cells have already evolved in the tumor?
Yeah, I think so. You're basically asking, like, has it been observed that immunotherapy can backfire? Is that your question?
Yes. Exactly. Yes.
I think so. In brain cancer and GBM, I think there are examples of like hyper progression, and there's a lot of research being done into stratifying patients and predicting which are going to benefit or not. That's sort of a different system than here. What I would say is in this model, we're more looking at metastasis. So we could think of it as maybe a particular metastasis in a patient might be accelerated, but other metastases in a patient might be diminished, right? So you could have sort of these counterbalancing effects in one single patient.
Okay. And does that depend on different types of cancers too?
Probably. That's a good question. Yeah, I'm not sure. This is definitely, yeah, I mean, yeah, I'm not really sure some of the details of that's a great question. In metastasis, I think it would be more relevant in metastasis in general. Just this interaction between metabolism and the immune system, but I'm not sure about different cancers. I have to think about that. Yeah, I don't know.
Okay, because I looked on Google right now, and immunotherapy has been prescribed for only few types of cancers, not all of them. So that's why it came to my mind.
Okay, that makes sense. Yeah, the treatments might not be approved for more than one cancer or more than a few cancers right now anyway. That's a great point.
Thanks.
I have one more question.
Sure.
If I have not interpreted it wrong, then the models you now discuss about, one factor was the environment is constant and the cells are evolving. But in case of metastatic cell, the cell has a fixed genotype or a phenotype, but it is now facing a different environment. So now the environment is changing, but the cellular context is the same. So have you tried modeling that?
Yeah, that's a great question. I love that question. So yeah, it's sort of related to the seed and soil concept.
Yes.
If the seed stays the same and you place it into different soils,
Yes.
Yeah, so I should be more explicit about that. I think that's a great point. What I'm sort of hinting at is that the soil may be changing due to its vascularization. So I see this vascular parameters as changing the soil. So obviously this is just looking at one tumor or one metastasis, but between metastases, it could be very different vascularization. So if it's well vascularized region that the tumor metastasizes into, very harsh soil versus very nice soil versus a very harsh soil.
Yes.
Yeah, I like that question a lot.
So in that case, what sort of assumptions or what kind of a biological data will you require to build a model?
Yeah, that's true. We haven't really looked at metastasis so in the same model. So we could build a model where you have like circulating cells looking at different mets, and it's in a single model, and there's cells circulating between them, and try to parameterize that. We do have things like PET scans, and that we could sort of scan a patient and then we can sort of pick up on some of these Warburg phenotypes and increased glycolysis, increased acid, things like that. So there's a lot of techniques that are used to map imaging onto these agent-based approaches, especially with the microenvironment, because we should be able to get some clues about the microenvironment through imaging. But that's three-dimensional. This model is two-dimensional. So yeah, it's complicated.
Okay. Thank you.
Yeah.
Okay, I think we have now we have extended by 30 minutes, and
That was great.
I think Jeffrey has some other plans too. So if anybody is not having any questions right now, we will like stop the question answer session. Anybody else wants to ask? Okay. So I think we are done with the questions, and thank you very much for this interactive session. And we are very much like, we liked the presentations and we understood the story, and it's really what we can call new way of looking into the things, new way of understanding developing the hypothesis, understanding the problems, and it will be we will love to have in future also. We will communicate with you if we have any if you are free and if you are willing to talk to us, then we will arrange some other talks as well. And thank you very much from our side, and yeah, have a nice day.
Thank you so much for inviting me. I look forward to continuing to interact with you all and trying to be helpful to you all. So definitely reach out to me. Okay. Thanks again for the invitation.
Thank you.
Thank you very much.
See you.
Yeah.
Thank you for
Bye.
the share.
Thank you.
Thank you very much. Thanks a lot.
Thank you, sir.